Hi, my name is Bridget Heffernan, and I'm the Senior Vice President of Events at the Historia Project. Today, I will be giving a presentation about Henrietta Lacks and her contributions to science. So, who was Henrietta Lacks? Henrietta Lacks, born Loretta Pleasant, was born on August 1st, 1920 in Baltimore, Maryland. Her mother died when she was 4, so her family moved in with her grandfather in Clover, Virginia until she got married at 21 when she moved back up to northern Maryland. Prior to her 5th pregnancy, Henrietta had felt a knot inside her, and this worry was confirmed when she experienced bleeding and a prominent lump on her cervix. She visited the doctor, who referred her to a gynecologist at Johns Hopkins Hospital in Baltimore, one of the few hospitals to treat poor African Americans at this time. A biopsy, a procedure in which the patient's tissue cells are removed for a microscopic examination, confirmed the presence of a cervical tumor that had gone undetected during her previous pregnancies. Henrietta underwent multiple radium treatments, which was standard for the time period. This radioactive treatment was thought to kill tumor cells after the cancer had spread to the bone. However, nowadays we have safer and more informed methods of treating cancer. The procedure involved stitching small glass tubes of radium in fabric pouches to the cervix. During the procedure, the surgeon extracted 2 small tissue samples, one from the cancerous tumor and one from the neighboring healthy cervical tissue. These samples were taken for physician George Gay, the head of tissue culture research at Johns Hopkins Hospital, so he could conduct cancer research. George Gay specifically needed cancer cells for this research because they were immortal, as cancer cells do not undergo apoptosis, aka programmed cell death. When a cell is damaged, it becomes inefficient and needs to be removed for another reason pertaining to the survival of the organism. Apoptosis is a normal occurrence in all of our bodies. For example, when we were playing outside and accidentally fall and get a scratch. When this happens, our cells send signals to each other to kill the inflamed cells so new cells can take their place and our skin can heal, a completely healthy and normal process. However, cancer cells do not undergo apoptosis, which means that if there are problems within the cell that would normally require them to die, they simply do not. Instead, they multiply, and this accumulation of immortal cells becomes a tumor. The samples of cancerous cells taken from Henrietta not only survived, but multiplied at an extraordinarily high rate. Other samples of cells taken from other patients had died quickly in Gay's laboratory, but hers proved different. These cells were nicknamed HeLa, after the first 2 letters of Henrietta's first and last names. She was unaware that the samples had been taken during her radium treatments. Today, HeLa cells are used to study the effects of toxins, drugs, hormones, and viruses on the growth of cancer cells without experimenting on humans. They have been used to test the effects of radiation and poisons, study the human genome, learn more about how viruses work, and played a crucial role in the development of the polio and the COVID-19 vaccines that have been administered to millions of people today. Sadly, Henrietta's health declined as cancer spread through her body, and she passed away on October 4th, 1951, at the age of 31. However, HeLa cells were used by multiple doctors for crucial medical research that we benefit from today. For example, you might have heard of Jonas Salk, the man behind the creation of the polio vaccine. Well, Jonas Salk actually used HeLa cells to develop it. Over 10,000 patents involving HeLa cells have been registered, and these cells were used to study disease and test how humans would react to new products and substances. Despite the massive medical advancements that HeLa cells contributed to, Henrietta's family remained unaware of this. Nor Henrietta or her family had received compensation for the use of her cells by biotech or pharmaceutical companies since they were harvested. The lack of credit has raised questions about the legality of using genetic materials without permission, as Henrietta did not consent to have her cells cloned and then sold. Luckily, in 2013, nearly 60 years after her death, the National Institute of Health granted the Lacks family control over how data of the HeLa cell genome would be used. 2 members of the Lacks family joined the NIH's HeLa cell genome data access working group, which reviewed researchers' applications for access to the HeLa sequence information. In honor of Black History Month, it is essential that we continue to acknowledge the priceless contributions that Black Americans have made to this country. Since Henrietta's death in 1951, the Lacks family has been fighting to receive the rightful compensation and credit for the use of HeLa cells. HeLa cells have played a critical role in the development of vaccines and medical research that all of us benefit from today, and we owe it all to Henrietta Lacks. Thank you for watching. I hope you all learned a little something about Henrietta Lacks today. I would also just like to thank my partner in creating this presentation, Belen, who did a lot of really great research. Thank you again for watching, and please contact the Historia Project if you have any questions or want to learn more. Thank you.